So in the past few years, I've been working on S100a7. This is a pro-inflammatory protein, and that drives the expression of a secondary protein that is Cpl A2, cytosolic phospholipase A2, and it modulates the tumor microenvironment of in metastatic breast cancers. So first of all, I would like to go over through the background and some of the hypotheses that we have in the mind. Triple negative breast cancer is one of the most aggressive form of breast cancers. So breast cancers can be classified into various subtypes based on their surface receptor expression. So out of them, TNBC is the most aggressive form and correlates with the poor prognosis and poor overall survival, as it metastasizes widespread in the body and too early. And we do not have established molecular targets for TNBC. So that's why there is a greater need to understand the molecular mechanisms that drive the aggressive behavior and metastasis to widespread in the body. So in order to develop better innovative therapies for against TNBC. So we and our lab, we mostly work on tumor suppressors and oncogenes. So S100a7 family belongs to one of the pro-inflammatory proteins, and inflammation has been associated with one of the hallmarks of cancer. So we were interested in elucidating more about this protein. So in the past, from our lab, we have published a few papers where we have shown S100a7 expression is really important for driving tumor growth and metastasis in breast cancer. And whereas, as opposed to its overexpression, its downregulation inhibits tumor growth and metastasis. So there was an interesting paper in 2017 in Nature Medicine where they have shown that S100A genes, its family, like one of the member, S100A7 gets amplified in subpopulation of breast cancer stem cells, including other family members of S100A. So we were intrigued about this finding, and we wanted to go dive in and investigate the whole signaling pathway and how we can use this as a novel molecular drug target. So we hypothesized that this is an inflammatory molecule and it might carry out down the lane some signaling pathways that are essential for tumor growth and metastasis, and it might modulate the tumor microenvironment because it's now it's emerging as a new drug target. So I would like to give you a brief overview about S100a7. This protein is also known as psoriasin because it is highly expressed in psoriasis disease. So it is the family member of S100 gene family that has many other S100 proteins, including S100a2, S100a4, 8, 9, 12, and so on. And this is a very small molecular weight protein, around 11.7 kDa. And these proteins are calcium binding proteins. Usually they exist as dimers under physiological conditions to carry out their signaling. And one of the most important aspects of these proteins are they are very small but chemoattractant. So they can attract inflammatory cells. And there was also one recent finding where they have shown this protein can be antimicrobial in nature as well. So the molecular mechanisms, how S100a7 promotes tumor growth and metastasis is not properly understood. And S100a7 is highly overexpressed in metastatic breast cancer, including TNBC, and promotes the expression of Cpl A2. So we looked up what is the downstream target since it's an inflammatory protein. So we looked at the downstream pathway and we found Cpl A2 might be the targets. And when we analyze it, it correlates with the expression of Cpl A2. However, the crosstalk between these two and the downstream signaling is poorly understood. So Cpl A2, as I mentioned before, it is a protein that plays an important part in inflammatory driven pathways and also in the biosynthesis of lipids, including prostaglandin E2, PGE2. So our lab in the previous years have developed a novel bi-transgenic conditional mouse model for S100a7. So S100a7 is the closest human homologue in mouse. So we have developed a conditional S100a7 overexpression mouse model, especially in the mammary gland, as we are interested in analyzing its role in epithelial S100a7 overexpression in breast cancer initiation and progression. So what we found is when we fed these mice with doxycycline, so this doxycycline can induce this tetracycline activator, and down the lane S100a7 gets produced. And we validated using Western blotting, where we have doxycycline, we see the expression, and where we do not see an expression. And this was further validated by IHC. You can see these the mammary glands where brown staining is positive for S100a7 expression. And once, so there was an interesting finding that was also we published in Cancer Research. So when we keep these mice for long under doxycycline, we see the induction of hyperplasia in the mammary gland. So you see this is carmine staining, this is S100a7 expression under doxycycline, and this is HNE, and this is cytokeratin staining. You can see whenever there is doxycycline, we see more S100a7 and hyperplasia. So we were interested in downstream, what are the interacting partners, as we do not have any inhibitor or neutralizing antibody for S100a7. Right now, we are in process to develop novel neutralizing antibodies against S100a7. So for that, we wanted to go into Cpl A2 and S100a7. If S100a7 is driving its effect through Cpl A2, it might be a probable or possible potential inhibitor to develop, as Cpl A2 inhibitors are already in clinical trials. So we analyzed public data sets and our own tissue microarrays for breast cancer. And we see S100a7 in the tumor, the staining is positive and very high. It's a secreted protein. That's why you see all over S100a7 signals. Compared to the adjacent normal, the expression is significantly high, and we quantitated in the graph. And we have analyzed other CHIN and CalDES datasets, GEN-T2 dataset, and SEEK dataset, and to analyze the expression of S100a7 in low grade and high grade. So in high grade, S100a7 expression is very high in breast cancer patients. So similarly, we did the analysis for Cpl A2. Cpl A2 was also very high in tumor compared to adjacent normal, and its expression was also high at the high grade tumors in breast cancer. And we did a Pearson correlation, Spearman correlation analysis using RTMA data for Cpl A2 and S100a7, and we see it's highly positively correlated. So their co-expression is highly correlated in breast cancer samples. So, and further, we wanted to analyze the survival. So how does these two proteins affect survival? So we analyze KM plotter dataset that's publicly available. And when we analyze both of the genes together, we see their higher expression correlates with poor survival. So next, we wanted to go into the signaling aspect of Cpl A2, how S100a7 is regulating Cpl A2. So S100 family, they mediate its effect. They are ligands. So they mediate their effects predominantly through two receptors. One is the TLR4 and the other is the Rage. So we analyzed the expression of TLR4 and its knockdown and its effect on S100 on Cpl A2, but we did not find any significant difference. So we moved on to the next receptor, that was Rage. When we analyzed the importance of Rage in enhancing Cpl A2 expression, so we see a significant difference. As you can see, we used GMB-2021. This is a human TNBC cell line where we have overexpressed S100a7. So here, when we overexpress S100a7, we see higher Cpl A2. And other than that, these 231 cells, they do not have S100a7. So as long as they have human S100a7, they make Cpl A2. So we picked up another cell line where basal level expression of S100a7 is already there. You can see that without the shRNA, you see S100a7 expression, and we knocked down S100a7. So, and then we followed Cpl A2 expression. So we see S100a7 has some role to regulate Cpl A2 expression down the lane through. So here in these cell lines. So we analyzed, further we wanted to analyze whether it's endogenous role of S100a7 or it's mediating through the receptor. So we used human recombinant S100a7 protein for 24-hour and 48-hour. These are the different concentrations. And we see when we treat with recombinant protein, we see increase in S100a7 and Cpl A2 protein once we treat them with recombinant S100a7. And to confirm the role of Rage in driving Cpl A2 expression, we used Rage specific siRNA, and in presence of Rage siRNA, you see Cpl A2 expression is also decreased. So that's how up till here we know that Rage is the receptor for S100a7 upregulating the expression of Cpl A2 downstream. So further we have, there is an established inhibitor for Rage receptor. So we use that also. Apart from the genetic aspect, we wanted to go for the inhibitory aspect so that we can further analyze down the lane. So this inhibitor also decreases in a dose dependent response at 20 micromolar. You can see this was for 24 hours when we treat these overexpressing 231 cells. So you see Cpl A2 expression is going down. So right now we know the information, whatever we have so far here is S100a7 is important, and it enhances its effect on metastasis and tumor growth via Cpl A2. So Cpl A2 also plays an important role in synthesis of PGE2. So we wanted to look at the level of PGE2 in these cells where we have S7 overexpressing 231 cells, and we have MD-MB-468 cells where we have knocked down S100a7. So in both these cell lines, we see similar results like in agreement to previous results. PGE2 levels were low when you don't have S100a7 or you do not have Cpl A2. So when we use Cpl A2 siRNA, we see lower production of PGE2. Similarly with the knockdown data. And we wanted to analyze the correlation between S100a7 and PGE2 levels. So we used breast cancer patient samples. We did ELISA for both of these proteins, and we see a positive correlation of S100a7 expression with PGE2 production. So further, we wanted to analyze the role of these important proteins in vivo. So what we did is we used an orthotopic model of S100a7 overexpressing 231 cells and the vector control cells. So here we have, we cultured them and we made spheroids, and these spheroids were injected in the mice, and down the lane when they have palpable tumors, we treat them with AACOCF3, that's a Cpl A2 inhibitor, for 4 weeks, and then we analyze tumor growth and metastasis. So when we treated them with I'm abbreviating this whole inhibitor as AF3. So when we treat them with vector control with vehicle control and AF3, we do not see much effect here. But when we use S100a7 overexpressing cells and we treat them with the inhibitor, you see a significant difference here. So S100a7 is mediating its effect through Cpl A2. So when there is no S7, even if you inhibit Cpl A2, there is no regression of tumors. And these are the tumor weight. They follow the tumor volume pattern. They are significantly low in the S100a7 overexpressing breast cancer cells treated with the inhibitor. And we analyzed the stemness also. We use these tumors and make single cell suspension and analyze them for CD44 high and CD24 low cells. So the results were, when we treat them with the inhibitor, we see a significant difference in reduction of the cancer stem cells in the tumor. And the difference was also high if you compared with the 231 vector and S7 231 overexpression. You can see that whenever you have S7, you have more cancer stem cells. And further, we analyzed the metastasis in the visceral organs. We see a significant difference in liver metastasis when you treat them with AF3. When we treated these mice with AF3, we see reduced metastasis in the liver, whereas in 231 cell lines that don't have S100a7, they do not have a significant difference between the two groups. We also wanted to analyze these tumors for their proliferation rate. So we did a Ki67 staining that represents its rate of proliferation. So highly proliferative cells were in S100a7 overexpression vehicle control, whereas if you look at the treatment group, we see significantly reduced number of Ki67 positive cells. So next was the previous data on orthotopic model was on immune compromised mice, so that we cannot evaluate the whole tumor microenvironment, T cells and B cells, as these immune compromised mice, they do not have functional T and B cells. So further, we have this, as I explained earlier, we have S100a7 overexpression mouse models. So we wanted to evaluate the effect of Cpl A2 inhibitor on MMTV implanted orthotopic breast cancer tumors. So MVT1 is a murine TNBC cell line. We implanted these cells in the mice, and once they develop palpable tumors, we add tetracycline, and so the one group has no tetracycline and the other group has tetracycline on, as we feed these mice with doxycycline. So TET off, TET on represents doxycycline, as doxycycline activates this tetracycline activator. So here we are referring TET off and TET on. So when we have the TET off system where we do not have S100a7 expression, so we do not see significant difference in the tumor growth. But when we have the TET on system where mouse S100a7 is overexpressed, so we see a significant difference when we treat these group with the AF3 inhibitor. So here clearly it indicates S100a7 is driving its effect through Cpl A2 to enhance metastasis and tumor progression. So we compared the tumor weight and tumor volume, like in previous results. We did see similar kind of thing here. When we have doxycycline fed mice, we see a significant difference of this inhibitor with the control group. Otherwise, we do not have a significant difference when the system is off. We also analyzed liver metastasis and lung metastasis. You can see huge lung nodules here. So in the on system, and when it's off, you see, or when we treat them with the inhibitor, you can see the lung nodules are very low. So we have quantified them, and we see a significant difference when we treat them the tetracycline on system with AF3, that's a Cpl A2 inhibitor. Further, we wanted to analyze these tumors for infiltration of different immune cells. So we have utilized a highly advanced multiplex imaging technique known as CODEX, stands for code detection by indexing. These studies were done at Stanford Core Facility in collaboration with Dr. Nidhi Bhutani and her group. So then in this technique, so this is the overlay how we make barcode. We use formalin fixed paraffin embedded tissue microarrays. Then we image them and scan them, and then we process them through algorithms. So what we found is when this is all for the TET on system. So when we treat with AF3, we see higher infiltration of CD8 cells and CD4 cells. You can see over here, and we also have higher stemness, that's the CD90 marker. So with these results, we see if we inhibit Cpl A2, there are no longer aggressive effects of S100a7 downstream. So these were the mouse data. So we wanted to, these were all orthotopic tumors, but since in breast cancer, we have availability of spontaneous, TNBC mouse models. So one of the mouse model is C3 tagged mouse model. So that develops spontaneous TNBC tumors on its own. So we cross this mice with the S100a7 overexpression mice. And when we have these C3 positive S100a7 mice fed on doxycycline, you can see the tumor numbers, tumor weight, and tumor volumes were much higher than the TET off system. So, and similarly, we see more metastasis in the lungs, and the nodes were bigger and in higher numbers in the TET on group, where we have more S100a7. So S100a7 aggravates tumor growth and metastasis in the spontaneous breast cancer mouse model as well. So this intrigued us to go into evaluate its clinical utility in breast cancer patients, since we do not have established molecular targets for TNBC. So we wanted to evaluate the whole system somewhere where we can have human implications. So for that, we utilized a humanized PDX mouse model. So how this is a schema, how we make humanized mouse model. So first we myeloablate these mice through radiations. and we inject human CD34 positive hematopoietic stem cells. And after a while, when these mice recover, we take some blood out and analyze human CD45 positive T cells. And once we confirm this data, these mouse have developed human T cells and then we inject these PDX. So we have also developed a human patient-derived TNBC PDX mouse model in our lab. So when we inject with the PDX, when we engraft, and once they have palpable tumors, we treat them with the inhibitor or vehicle control. And once they reach endpoints, we sacrifice them and analyze the visceral organs for any sign of metastasis. So this is the PDX tissue where we have high expression of S100a7 and Cpl A2. So when we treat them with vehicle control, the tumors are much bigger and aggressive in behavior. And when we treat them with a Cpl A2 inhibitor, tumor regresses significantly. So these are some of the graphs that quantitate the tumor volume and tumor weight. So further, we wanted to look at the tumors for their Ki67 positive cells. As you can see in vehicle control, they are highly proliferative and positive for Ki67 staining. As opposed to AF3 treated group, they have very low staining of Ki67 positive cells. And when we looked at the lungs, we see a significantly higher metastasis in the lung compared to the AF3 inhibitor treated group. So as I mentioned before, S100a7 protein also enhances cancer stemness. So we wanted to look at these tumor cells. When we treat them with the inhibitor, do we see a reduction in cancer stem cell population? When we analyze these tumors for CD44 high positive cells, so we see a significant reduction of CD44 positive cells, CD24 low and CD44 high cells. So you can look at this quadrant, that's 23 in the vehicle control and it's 4 like significantly low. So it reduces the cancer stem cell population in the tumor. So further, when we treat with this inhibitor and we see shrinkage of tumor and low metastasis, so what is driving it? Are these tumor cells not dividing or going into senescence when we treat them with the inhibitor, or they are going through the process of apoptosis, cell death? When we analyze these tumors for cleave caspase 3 staining, so cleave caspase 3 is a late apoptotic marker. When cells are positive for this, that means cells are going to die by a programmed cell death pathway. So these cleave caspase 3 positive cells were very high in AF3 compared to the vehicle control. So that clearly shows here, when we treat with the inhibitor, it induces apoptosis in tumor cells, and that's how we see a shrinkage of tumor. So further, we wanted to look at these humanized mouse model. So what are the expression of infiltration and recruitment of M2 TAMs? So TAMs stands for tumor-associated macrophages, and these macrophages supports tumor growth and metastasis. So when we analyze the whole percent TAM population in the tumors, we see it's significantly reduced in the AF3, and TAMs can be classified. Majorly TAMs are M2, we call them TAMs, but macrophages are of two main subtypes. One is M1 and the other is M2. So M2 promotes tumor growth. So we wanted to look at which population is high. So when we analyzed M1, we did not see higher significant difference between the two groups, but when we looked at M2 population, that's here CD163 positive cells, we see the population is significantly lower in AF3. So when the population is low, that means it can no longer support tumor growth and metastasis. And when we analyzed the CD4 and CD8 T cells, so there was no significant difference in CD8 positive T cells, whereas we see a significant difference in CD4 positive cells. So we looked at their surface expression for any checkpoint expression or any sign of activation. So when we analyze Cpl A4 positive immune checkpoint, we see CD4 positive CTLA4 is highly expressed on these vehicle control cells. And when you treat them with the inhibitor, the CTLA4A expression goes down. So that's a checkpoint that inhibits the function of CD4 T cells. CD4 T cells are essential for activating CD8 cells. They carry out predominantly the helper functions in the tumor microenvironment, and sometimes they can also be cytotoxic, so that we have to look. So far we see the percentage of CTLA4 positive CD4 T tumor infiltrating lymphocytes is significantly low in AF3 treated mice. So we further analyze, as we know, we have humanized mice model data with where M2 TAMs were low. So we wanted to go further and look at the human breast cancer patient samples. So we utilized tissue microarray that we utilized for S100a7 and Cpl A2 expression. And when we stain them for M2 TAMs, CD163 positive cells, we see higher positive cells in high grade tumor. And you can look at the graph where you have high grade tumors, you have very high expression of M2 TAMs in the tumor microenvironment. So when we draw a Spearman correlation in between S100a7 and Cpl A2, so we see a positive correlation of M2 TAMs with Cpl A2 and S100a7. Further, we looked at a publicly available data set that is known as timer. So where we looked at the M2 population is also correlated with the expression of Cpl A2. So here you can see it's positively 0.4 is like highly positively correlated. So with all these data, we were able to say S100a7 down the lane activates Cpl A2 to play an important role in enhancing breast cancer growth and metastasis. And Cpl A2 S100a7 are also important for recruiting M2 TAMs in the tumor microenvironment, where it supports the tumor growth and metastasis. So the studies have also shown PGE2 levels are high, then there is more recruitment of TAMs. And we also see with our in vitro experiment data, PGE2 levels were high when you have high S100a7 and Cpl A2. So, and pharmacological inhibition of Cpl A2 protein reduces tumor growth and metastasis in various TNBC or breast cancer preclinical mouse models, including C3-TAC TNBC mouse model and humanized PDX mouse model. When we target Cpl A2, it attenuates S100a7 mediated downstream effects for promoting growth and metastasis. So we believe it's a novel strategy to develop precision medicine against metastatic breast cancers where we have limited therapeutic options. And pharmacological inhibition of the signaling axis might be a better approach to abrogate the effects, immunosuppressive effects in the tumor microenvironment driven by this complex of these two proteins. So overall, our studies using in vitro assays and mouse models and patient samples suggest novel roles of S100a7 and Cpl A2 signaling, oncogenic and inflammatory signaling for enhancing breast cancer growth and regulating the immunosuppressive tumor microenvironment. I would like to thank my PI, Professor Ramesh Kanju, my lab mates Dinesh, Sanjay, and we also like to acknowledge Bhutani lab at Stanford that helped us with the analysis of different immune cells, and the core facility at Stanford. On NIH funding, we acknowledge the funding agencies, NIH, DoD, and Peltonia. Thank you. I'm happy to take any questions.